Formation of pollen. The reproductive organs of angiosperm plants are called flowers. Flowers consist of several sections interwoven around an axis and held on the tip of stems of varying lengths. These are stem, peduncle, receptacle, sepal, petal, stamen, and carpel. Angiosperms have male organs consisting of two parts, anther and filaments. The cross-section of the other displays four obvious compartments. Each compartment contains pollen sacs. Within each pollen sac, there are many parent cells containing diploid, that is 2n, number of chromosomes. Each parent cell produces 4 haploid N daughter cells called microspores through meiosis. Through mitosis, each microspore produces pollen grains containing 2 nuclei. Each pollen grain contains 2 genetically identical haploid N nuclei. Of these, one vegetative nucleus provides for the formation of the pollen tube, whereas the other generative nucleus divides through mitosis to produce sperm nuclei, which has the role of fertilization. Pollen will vary according to the plant color, shape, and structure. Pollen grains are surrounded by a double membrane layer. The outer membrane may vary in structure, such as spikes, pores, or patterns on its surface. The inner membrane is smooth. The inner membrane is live, while the outer membrane is dead. The maturing pollen sacs explode to release and disperse the pollen sacs into the environment.
Pollination is the act of transferring pollen grains from the male anther of a flower to the female stigma. Seeds can only be produced when pollen is transferred between flowers of the same species. There are 2 types of pollination. First, self-pollination. Second, cross-pollination. Self-pollination occurs when pollen grains fall directly from anther into the stigma of the flower. This process is quite simple and fast, which leads to a reduction in genetic diversity as the sperm and egg cells of the flower belong to same plant. Cross-pollination, the transfer of pollen grains from the anther of the flower into the stigma of another flower. This method increases in genetic diversity, as different flowers will share and combine their genetic material to create unique offspring. Types of cross-pollination. Pollination by wind, anemophily. Only a few flowers that use wind pollination. Pollination by animals, zoophily. Animals play an important role in plant reproduction. Artificial pollination is done by human beings. The process of fertilization in plants will take place when the sperm cells will reach the ovary and egg cells. The seed is then released from the parent plant and making it able to grow into a plant and continue the reproductive cycle with the use of the pollination method.
Flowering plants undergo a unique reproductive process where there are 2 fertilization events. This double fertilization event occurs between the male reproductive organ, the male gametophyte, and the female reproductive organ, the female gametophyte. Before the fertilization event can occur, the ovule has to undergo some changes. At present, the ovule contains one reproductive cell, known as the megaspore or mother cell. This cell is diploid and undergoes meiosis, producing 4 haploid megaspores. In the majority of species, 3 of these megaspores degenerate, leaving only one surviving megaspore. This surviving megaspore expands and undergoes 3 rounds of mitosis to produce 8 haploid nuclei. As the nuclei have not developed any individual division, they initially share the same cytoplasm. This complete structure is known as the embryo sac. Within the established embryo sac, cell walls begin to form between most of the nuclei. 3 cells named antipodal cells form opposite the opening of the ovule, known as the micropyle. Another 3 cells form above the micropyle. 2 of these are synergids, and the other is the egg cell. This leaves 2 nuclei in the center of the ovule. These central nuclei remain together in one large cell. It is the egg cell and this central nucleate cell which will eventually become part of the double fertilization event. In order for the double fertilization event to occur, the male gametes, the sperm, must travel from the anther to the embryo sac within the female reproductive organ. The pollen grain contains 2 main cells. A cell named the tube cell makes up the bulk of the pollen grain, and the sperm cell, which at this stage is known as the generative cell. To reach the embryo sac, a pollen grain must land on the stigma. Once landed, it begins to germinate. The tube cell forms a long structure down the style and into the ovary. The generative cell travels behind the tube cell nucleus. Once near the ovary, it divides by mitosis to produce 2 haploid sperm cells. The pollen tube reaches the micropyle and releases the sperm cells into the embryo sac. One of the 2 sperm cells fertilizes the egg cell. This produces a diploid zygote, which will become the embryo. The other sperm cell moves up and fuses with both of the central nuclei, forming a triploid cell. This unusual triploid cell develops into an endosperm and serves as the embryo's food supply during early development. It is only angiosperms, flowering plants, which have this double fertilization characteristic, where a diploid zygote and a triploid endosperm form. Gymnosperms, pines, tracheophytes, ferns, and non-tracheophytes, mosses, lack this double fertilization feature.